In cattle, the bull will deposit 5 to 15 billion sperm in the anterior vagina and fornix vagina of the cow. Addition of seminal fluid to the sperm serves to activate the sperm motility and also add a coating to the sperm to prevent fertilization until they move through the reproductive tract. Note that the contraction of the sperm tail causes rotation of the paddle-shaped head. Sperm move into the cervical os through the clear, stringy mucus that originates from the cervix. Sperm that swim into the deep cervical crypts move into the uterine body. The cervix serves as a sperm reservoir and barrier in cattle to restrict the numbers of sperm in the oviduct. Estrogen from the Graafian follicle stimulates myometrial contractions which propel the sperm through the uterine horns to the uterotubal junction. Sperm numbers entering the oviduct at one time are restricted by the valve-shaped uterotubal junction, but less so in cattle compared to the pig and horse. Only about 100 sperm may be present in the vicinity of the ovum following ovulation at any one time. Sperm move through the isthmus and may bind the cilia of the oviductal epithelium of the ampullary isthmic region. Binding to the epithelium may assist in keeping sperm close to the site of fertilization and allowing them to be released when they become capacitated. Capacitated sperm will bind to the zona pellucida of the egg by attaching its acrosomal region to the sperm receptors ZP3. Attachment induces the acrosome reaction to cause vesiculation through fusion of the plasma membrane and the outer acrosomal membrane to release enzymes allowing penetration of the zona. The acrosome-reacted sperm moves into the peripatelin space, and its post-acrosomal region binds and fuses with the oolemma of the egg. Fusion causes egg activation through rapid calcium release, which quickly causes fusion of the enzyme-containing cortical granules with the egg plasma membrane. Release of the enzymes from the cortical granules induces the vitelline and zonal block to polyspermy. Fusion of the sperm also initiates completion of meiosis II within the ovum, which releases the second polar body into the perivitelline space, and the haploid female pronucleus is formed. The entire sperm head and its tail come into the ooplasm. The head will decondense, the male pronucleus is formed, and moves into the middle of the ovum with the female pronucleus. The centriole of the sperm tail also moves to form the spindle fibers for cell division. The nuclear envelopes of the male and female pronucleus break down, the chromosomes line up, duplicate, the mitotic spindle forms, and the first cell division occurs. The fertilized egg will undergo several cleavage divisions. The embryo enters the uterus about the 8-cell stage and continues dividing with formation of a morula that will develop tight junctions within its outside cell layer, termed compaction. Compaction of the morula sets up cell polarity for fluid accumulation in the center of the embryo to form the blastocyst. Influx of intracellular fluid forms the blastocele, with the outer cells destined to differentiate into trophoblasts and further into placenta, and the inner cells forming the inner cell mass destined to form the embryo. Enzymatic activity and the thinning of the zona of the blastocyst leads to hatching from the zona pellucida when the embryo is in the uterus.